Welcome to another episode of Histology Labs. This is Dr. Bison, EM. Today we're looking at epithelia 2, which are exocrine glands. This is Histology Practical 4. Without wasting much time, let's get into this practical session. So on the introduction, you have to know that epithelial cells make up glands, being either exocrine, which possess ducts, or endocrine glands, which are ductless. Glandular epithelia, which is also known as glandular tissue, refers to a type of epithelial tissue involved in the production and release of different secretory products. Glandular epithelia is arranged into structures known as glands, which can either be a single cell or a group of cells specialized in producing and releasing a specific substance, such as sweat, saliva, breast milk, digestive enzymes and hormones, and many other substances are produced by glands. Now, exocrine glands produce and secrete a large variety of fluids that are important for digestion, firstly, lubrication, lactation, and thermoregulation. Examples of exocrine glands include sweat glands, salivary glands, lacrimal glands, those glands that produce tears, and mammary glands. These glands are composed of layers of secretory epithelial cells that absorb water, ions, and hydrophilic macromolecules from blood and secrete them onto epithelial surfaces through a duct system. Now, this diagram is a schematic drawing of an exocrine gland. So an exocrine gland has got this duct portion here. So these are ductal cells. This is a conducting portion. And then it has these acini cells. So this is the gland. The glands will produce among many other things, sodium, chloride, and water. And after they are produced from the acini cells, the product of the glands is going to go through the conducting system, through the ductal cells, and it will reach the surface epithelia of the cells where it is going. The secretory portion of the sweat gland is this one here. So this is the secretory portion of the sweat gland. And it is coiled, as you can see. So we say it is a simple coiled gland. Okay, this is the conducting portion, and then this is the secretory portion. So how is glandular epithelia formed? Glandular epithelia is formed during fetal life, in utero life, when there is fetal development in the uterus, by the development of epithelial cells into the connective tissue underlying the epithelium. So initially, all glands are connected to the surface by a channel called a duct. However, later on, each gland will differentiate into exocrine or endocrine according to the presence or absence of ducts, respectively. So this is to say that exocrine glands have got ducts and endocrine glands have no ducts. Now, what are compound alveolar glands? So secretory glands can be categorized according to the shape of the secretory unit. Secretory units shaped as a tube are referred to as tubular, whereas spherical units are referred to as alveolar or acini glands. Exocrine glands can also be composed of both tubular or alveolar secretory units. In this case, they are referred to as tubular-alveolar glands or tubulo-acinar glands. They can also be categorized according to whether their duct is branched or not. An unbranched duct is referred to as a simple gland, whereas a branched duct is also known as a compound gland. An example of a simple gland is a sweat gland, whereas the pancreas is an example of a compound gland. So this is a schematic diagram that is showing us. This is a tubular gland. Simple. This is a branched tubular. This is a coiled tubular. and this is the branched alveoli. This is a compound tubular gland, and this is a compound alveolar gland. So it is very important for you to orient yourselves to this classification of glands. Now, in this session today, what are our objectives? So number one, to classify glands on the basis of their morphology and types of secretion. Secondly, to describe the structures of different types of exocrine glands. Thirdly, to enumerate factors that stimulate or inhibit secretions by various glands. So what I'm going to tell you in this video is exactly what you're going to draw in your reports. Share this video with everyone in the classes. So let's start with our first slides. Let's start with simple tubular glands with unicellular glands. Okay, so this slide is showing the ileum. Remember, last time when we looked at epithelial 1, we said in the ileum, this is what we call our plica secularis. And then these are our villi. Okay, so the villi have got these glands here. These glands are called simple tubular glands. So what happens is that when you have a villi and another villi, between the two villi, there's a crypt called crypt of Lebucan. So those crypts will be continuous with epithelia and they will form glands. So this is at low power. This is a gland here. It is lined by epithelial tissue. This is a gland. It's lined by epithelial tissue. So they're cut in different sections. So this is cross section. This is transverse. Okay. So this is at low power. And then this is at high power. So only draw these glands at high power. So this is your gland here. And this is your glands. So the type of epithelia that are found in these glands are columnar cells. So as you can see, these are columnar cells. So these are columnar cells that are surrounding these glands. So at high power, just draw one gland here and this gland here. That's all in the ileum. Okay. So between adjacent villi, straight tubular glands look like tubes or pits with continuous epithelia. Intestinal crypts of Lebucan at the base look for red granules, which are Punnett cells. Now, what is the function of Punnett cells? So when you draw these glands, you should look for Punnett cells, label Punnett cells, and only draw at high power. Only draw at high power. Okay. So this is the same. So this is an intestinal villi. This is an intestinal villi. Between two villi, villi number 1, villi number 2, this is what we call crypt of Lebucan. So the crypt of Lebucan are continuous with epithelial cells and they form a gland. These glands have got simple columnar cells, and at the base of this gland there are Punnett cells. So I want you, after you draw these glands, so draw this gland here. This is your Punnett cell. Draw this gland, Punnett cell. Label them, and on the side write the function of the Punnett cells. Only draw this gland at high power. Let's move on. Let's go to the uterus. At the uterus, so look for tubular glands extending from the lumen of the organ, lined with simple columnar epithelia. Some may be branched, so only draw at high power. And then also look for the other slide. Will have a secreting mucosa. So the uterus is going to be at two stages. One will be a non-secretory uterus, and then the other slide, which is slide 88, will be a secretory uterus. So the difference between a secretory uterus and a non-secretory uterus is that the glands of the secretory uterus are tortuous and they're dilated. Okay. And then also answer this question after you draw. What is the other name for the secretory phase of the uterus? So this is a non-secretory uterus. So what you can see here, this is a gland here, this is a gland, 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 this is a gland. So in a non-secretory state, the uterus is the glands are very small. So these are glands, this is a gland, it is lined by simple columnar epithelia, this is a gland, this is a gland. So this is the diagram that you are drawing, only at high power. Okay. All right. So I'm looking for another uterus. We'll find it. So draw the uterus at non-secretory and also draw the uterus at secretory. As we go on, the slide for secretory will be found. Okay, so the scalp, you should identify sweat glands. Sweat glands are coiled tubular glands secreting by merocrine methods. Merocrine methods is where your secretory product is released by exocytosis. Okay. So the coiled secretory parts of the sweat glands are lined by simple or pseudostratified columnar epithelia, with the secretory duct which turns more darkly with a different epithelium. So only draw at high power for this. Only draw at high power for this. Okay. So this is a scalp. Look for the sweat gland, only at high power. So this is a sweat gland here. So this one here, the sweat gland. This is the sweat gland. This is the sweat gland. So only draw 3. So draw this one here, this one here, and this one here. Choose any. So this one here. So choose any 3, draw and label. Okay. So this one here are in cross section. So this one here are sweat glands. So this slide shows you sebaceous glands. So this is a sebaceous gland. A sebaceous gland is usually attached to the hair follicle, and it is not straight like a mucus, like a sweat gland. So both a sweat gland and a sebaceous gland are found under skin. So for the sebaceous glands, they're usually attached to the hair follicle, into which they empty. So observe epithelial capsule surrounding the gland with changing in size of the cells. Then pay attention to the frothy nature of the cytoplasm resulting from dissolved lipid droplets. These are circular glands which secrete by holocrine. So in holocrine secretion, there is destruction of the whole cell to get the product of the gland. So only draw at high power. This is a high power. This is a sebaceous gland. So draw all of this sebaceous gland. So this is the conducting portion here. And then this is the gland, the acini portion. So draw these cells in the gland and show the conducting portion as well. Okay. Now let's go to compound alveolar glands. An example is the breast mammary gland. So on this slide, you should note glands that are secreting and therefore alveolar are dilated with homogeneous pink-stained material. So the alveoli are lined with simple cuboidal epithelium whose lumen is uneven because glands secrete by apocrine. So the glands in the breast secrete by apocrine secretion. Identify the ducts, observe what their epithelia is similar. Observe that their epithelia is similar to that lining the ducts of sweat glands. Then identify what type of epithelia they are. Only draw at high power. Now let's look at this structure, see what we're talking about. So this is the breast at low power. So this is a lobule here. This is a lobule. This is an interlobular duct. So it's a duct that is between two lobules. So it's an interlobular duct. But inside the lobule, you see that there are these ducts here. There's a duct here, there's a duct here, there's the duct here. The ducts inside of the lobule are called intralobular ducts. So ducts inside of the lobule are intralobular. Ducts between two lobules are interlobular ducts. So this is what you are drawing. You're drawing this and you're showing these intralobular ducts. So these are in cross section. These are intralobular ducts, intralobular ducts. Draw about 4. 1, 2, 3, and 4. And then these red dots here that you're seeing, dot, these are blood vessels. And if you can see this colloid material inside of the intralobular ducts, this is mucus that is produced. So draw this mammary glands, show the intralobular ducts, show the type of epithelia that are surrounding this duct, and show the colloid material inside of the intralobular ducts. Draw and label. So breast histology, this is the same thing you're drawing. Okay, so this is the basement membrane. Then you have the myoepithelial cells, the luminal epithelial cells, and then these are the secretions. Okay, so this is what you're going to draw. This is an intralobule, intralobular duct. Okay, intralobular duct. That's where you find the colloid material, which is mucus that is secreted by the glandular epithelial cells that are found in the breast. Okay. Then slide 40, it shows Tubule-acinar glands that are serous and mucous acini. So identify, compare, and contrast the two types of acini labeled at high power, both secreting by Mallory methods. So draw a mucus secretory unit with a demi-lumen at high power. Identify interlobular and larger identify intralobular ducts and larger interlobular ducts. So this is we were just talking about it. It's in the breast. This is what this slide is talking about. Okay. So this is a sublingual gland. So the sublingual gland at low power looks like this. So you have a lobule here, and there's a duct here. This duct between these two lobules is called an interlobular duct. And inside of a lobule, you've got ducts as well, called intralobular ducts. Okay. Now, this sublingual gland is a serous mucous gland. So these portions that are stained maroon here, these are serous portions, and these that are stained white are mucous portions. So it's a serous mucous gland. It's a mixed gland. Okay. So this slide shows the interlobular duct. Intralobular duct. Okay, there's a mistake here. Intralobular ducts, because this here is a duct. It is found inside of a lobe. So it's an intralobular duct. Okay, this intralobular duct is lined by simple cuboidal. It's lined by simple cuboidal epithelia. Okay, so these here are serous. And then these ones that are staining pale here or white are mucous portions of the sublingual duct. So you're supposed to name all of these. So name the duct, name the serous portion, name the mucous portion, draw and label. Okay, so this duct diagram also shows the same things. Okay, so 3 serous acini, 6 mucous tubule, 4 serous demilunae. Same thing. Okay, this is the end of the lab. It's very, very short and it's very easy. The only thing that is missing here is a secretory uterus. So look for a secretory uterus, show it as well, and make sure that the glands in a secretory uterus, you draw them at high power, and the glands should be bigger than the glands that are in a non-secretory uterus. For future reference and for further study of this topic, go to Janquilla chapter 4 and study. I'll see you in the next video. This is Dr. Bison EM.